Hi everyone, thank you so much for joining us today at the LabRoots webinar series. My name is Manju Kang. I'm a senior scientist at the Antibody Drug Discovery Department at GenScript ProBio. Today I'm going to talk to you about the accelerated COVID-19 antibody discovery with ProSpeed single B-cell technology. First, I'm going to talk to you about the background of COVID-19, and next I'm going to present you with some of our newest data results for the antibody discovery for COVID-19. And lastly, I'm going to focus on the single-cell screening for COVID-19. COVID-19 has developed into a global pandemic that's affecting thousands, if not millions, of lives globally. By March 31st, the total confirmed cases reported by the SCCE at Johns Hopkins reaches over 784,000. And there are tremendous resources and efforts that are being poured into developing either the vaccination for protecting the general population over the long run or therapeutic agents to treat infected individuals. There are 7 different major drug targets that can be used for COVID-19. The one that's probably the most prominent or the most explored is the spike protein, which is on the surface of the viral particle, and it's responsible for the interaction with the ACE2 receptor on the host cell surface for the subsequent viral entry. And the other drug targets include the envelope protein, the membrane protein, the protease, the nucleocapsid protein, the hemagglutinin esterase, and the helicase. And there are also 16 non-structural proteins that can be targeted as well. But like I mentioned again, the spike protein is probably the ones that's being the most explored. So at GenScript, we have 3 different major antibody discovery platforms, which are Hybridoma, the antibody library, and we have both the human naive library. The human synthetic library is being developed right now, and then also the llama naive library for generating SDAB, or the single domain antibody. And our newest addition for our ABD platforms is the single B-cell screening platform, adopting the Beacon platform from Berkeley Lights. And we are utilizing all of our platforms to screen for desirable leads against COVID-19, which have the following 3 characteristics. The first is the ability to bind to spike protein, and the second is to block S protein and ACE2 interaction, and lastly, or ultimately, to neutralize or inhibit the COVID-19 infectivity. And on this slide, I'm presenting you some of our newest results from the phage display screening against COVID-19. The target that we're using is RBD of S1 protein, or the receptor binding domain of S1 protein, and we're screening against both our human naive library and llama naive library. The unique binder that we got for human naive library is over 70, and then we have about 48 SDAB screened from the llama naive library. Here, what I'm showing you is the FACS result for some of the binders that we're characterizing in vitrally. First, we did a RBD, or the dose response for the EC50 of RBD binding on the ACE2 or the ACE2 receptor expressed on the stable cell line. And what you can see right here is the partial blocking of the interaction or the binding between the RBD and ACE2 on stable cell lines. I'm highlighting some of the more interesting results. We have one lead with a KD at the nanomolar range that has a very significant or substantial blocking effect as well. And some of the things that I want to point out is we do have some binders that has a pretty good blocking effect, but on the ELISA results for the binding, it was not the best. It was not our best binder, but it shows a very good blocking effect, which is in concordance with some of the newest results from other I guess research or people as well, where the blocking effects that you might be looking for may not be coming from your best binder. So that's just something to keep in mind. So moving on, I'm going to focus on the single-cell screening for COVID-19 to give you an overview of our GenScript ProSpeed system. From immunization to recombinant MAB production, the shortest timeline that we can achieve is 1 to 1.5 months with express immunization, which takes about 2 weeks. The general workflow involves after immunization, the B cell will be harvested and enriched for plasma cell, and those B cell subset will be imported or penned into the chip that's used for screening, and with various in-chip assays including specificity, affinity, and functionality. And after we analyze the results and pick our desirable lead, the single B cell will be exported into 96 well plates. And follow that is the cDNA synthesis and sequencing and recombinant MAB production. And what I want to point out right here is from B cell harvest to exporting the single B cell that you picked out, this process can be finished within less than a day. And immediately you can see how valuable this platform can be for developing antibody leads that are extremely time sensitive. And this is a fully automated, efficient, and comprehensive platform to really accelerate your antibody discovery. The general workflow involves 3 major steps. First is to import your sample, and then next is to assay. And here we're showing you the assay can be done in a multiplex fashion where we can do multiple assays at the same time or have different results or different readouts for one singular assay run, and after that is exporting your cell of interest into 96 well plate for downstream single cell sequencing. So here I'm going to show you a little video of how exactly each process takes place. So the penning is where the OEP or the optoelectrode positioning system, where you can see as the little cages that's identifying and directing single cells into the nano pen, or the little room, or I guess the segregation that can be achieved at the single cell level. So each cell can be directed in a single pen for single cell isolation. So this is also a reason why the importing is called penning. As you can see again, the cells are being identified and directed or being moved into the bottom of the pens to achieve single cell isolation. And here the next part is the assay. So I'm going to play the little video again for you, and what you're seeing right here is the signal that's being detected as what we call a fluorescent bloom, where so if you look at the specific pen, you're seeing the signal that's generated or accumulated over time at the opening of each pen. So what that means is that at this particular pen, you have a plasma cell that's secreting antibody of your interest, and it's being detected by your assay system over this area or in a channel. And the signal can be very sensitive and also robust. The detection, it can be done within minutes, and it's highly reliable because it's basically taking a multiple time points or data points over time. And again, the multiplex assay can be achieved by running multiple assays sequentially or simultaneously. And lastly is exporting your cell of interest for downstream sequencing. Oops, sorry. So what you can see right here is you have the OEP or the optoelectrode positioning system exporting or directing the cells out of the pen. So what you're seeing right here is the movement of the little plasma cell being directed out of the pen into the channel, where it's going to be flushed into a single a single well in a 96 well plate. And the plate can be stored at -80 before sequencing. So after introducing you to the general workflow of how the system works for our single B cell screening platform, you might have some idea of why is the single B cell screening faster and even more robust with just one cell. So just to break it down for you and have a little comparison with the Hybridoma system, or what you would do for ELISA screening. So the media volume per pen, or the nano pen on the chip for the screening for single B cell is 1 nanoliter. And if you have a total cell number as being 1 cell, the concentration for your screening system is 1 cell per 1 nanoliter. But your effective concentration, and here what I did is just to convert the unit, or the unit for my media volume, to 1 mil. To get my effective concentration, which is equal to 10 to the 6 cells per mil. And if you compare that to Hybridoma, where media volume is 100 microliter, and the total cell you need to examine, or you need to wait for to grow up, is about 10 to the 4 cells per well, then your concentration for that system is 10 to the 4th cells per 100 microliter, which equals to 10 to the 5th cells per mil. And you can see right here, there's actually a 10-fold increase in the single B cell screening system compared to the Hybridoma system, which means you have increased robustness and also sensitivity, which is also time. It saves you time for getting an assay readout, because first of all, you don't have to wait for the cells to grow up to a certain density to allow you to detect your results. And this means that the single B cell screening can be detected, or the assay you can generate the result within minutes, while Hybridoma or ELISA, you have to wait for days to get your results. To summarize our platform, the OEP, or the optoelectrode positioning system, is used for the precise movement or direction of single cell into the nanopens, or to achieve single cell segregation. And the whole system is, after you import the cell, is completely automated from importing, screening to export, and this can reduce the chance to introduce human error with less human handling time. And then also the entire process is recorded digitally for quality control with both image and video. And the efficiency, as we just showed you, the screening for Hybridoma typically takes about 3 months, because there's multiple rounds of screening from the parental clone to the subclone, and you have to wait for the cells to grow up. But with single B cell, the ProSpeed platform, this whole process can be done within a day, or is essentially less than a day. The resolution, and here what we mean by that is your ability to access the vast antibody repertoire at the single cell scale, which is about 100-fold, or at least 100-fold increase from Hybridoma, because your fusion efficiency is typically 1 out of 1,000 B cells that can actually form a Hybridoma for you to screen. So at that step, you are losing a lot of the B cells due to the limitation of your fusion efficiency. And lastly, the versatility and flexibility. This system can be used for in-depth characterization, including affinities, specificity, and functionality for your B cell before you even export it for sequencing. Okay. So to give you an overview of the antibody screening process against COVID-19, if we're using the Hybridoma or phage display library system, or the platform, the initial screening will be used for the target discovery or lead generation, and follows that is the lead identification and optimization, which can take weeks to months, and that includes characterizing or validating the binding and blocking activity using ELISA or FACS, and characterizing the virus neutralization effect. And here what we developed is a pseudovirus system for COVID-19. And next you have to you have to validate the virus neutralization effect with the real COVID-19, and follows that is the in vivo functionality or animal studies before you move on to the clinical study or to even worry about developability. So the process is very arduous and very it's a very long process. But with ProSpeed single B cell screening, what we're trying to achieve is to combine the initial screening, the binding and blocking activity, and then also to look for virus that, or to look for antibody lead that has a neutralizing effect using the pseudovirus, the COVID-19 pseudovirus system on chip, or during the screening process, which can be done with less than a day before we move on to the identification or the optimization and validation with the real COVID-19, and then also the animal studies. So for the single B cell screening, we also have been doing some in-house antibody discovery. We have screened for a total of 613 binders, and within those 316, we were able to identify about 93 binders that have a ligand blocking effect. And with both of the assays, it's still, the screening process is still completed within 1 day. And with and here I have a little schematic to show you the experimental design and how we design our screening. So essentially what you have is we have a RBD, or the receptor binding domain of the S1 protein conjugated to a bead, and the bead is used to capture the secreted antibody. So if you have a secreted antibody that's specific to RBD protein or the RBD S1 protein, it will be captured by the RBD bead, and then you add a fluorescent secondary antibody for the detection of the correct specificity. And for the ligand blocker assay, what we had is the target cell line that's over expressing the ACE2 receptor, which is responsible for the interaction with the virus and then the viral entry. And again, so if we have a secreted antibody that has a blocking effect, what we're trying to see right here is essentially a negative result for the RBD, which is fluorescently tagged on the stable cell line. So by that I mean, if you have a secreted antibody that can block the interaction between the RBD or the binding of the RBD and our target cell line, we would not see the fluorescent signal that's generated from the RBD protein. And here again, you have the secondary antibody that's used for the detection of the secreted antibody. And what other assays that we're trying to develop is the pseudovirus, the COVID-19 pseudovirus assay. The pseudovirus is what we have right here is to replace the envelope glycoprotein of lentiviral vector by the COVID-19 spike protein. And here what I'm showing you is some activation data where we have different doses of pseudovirus, and you can see the luminescence signal from the infected cells with different doses of pseudovirus. So to verify the pseudovirus infectivity of our target cell line. And we also have done a neutralization assay where we have a positive control or positive antibody that was that can be used to successfully block of the effect of the infectivity of pseudovirus. So the design of our experimental approach again in the schematic is showing that instead of using a ligand or the RBD protein or the ligand the ligand for the ACE2, which is the RBD or of the S1 protein, we can replace that with the GFP expressing COVID-19 pseudovirus. So the idea is that if you have an antibody that's secreted from your plasma cell or from your cell of interest to block the effect or to block the pseudovirus infectivity of the target cell line, then we identify an antibody with the neutralizing effect. So, and this experiment can be done on chip as well. So aside from the different assays that can be used to develop antibodies against COVID-19, we can also accommodate different sample species origin. So what I just introduced you before is our typical workflow from immunized animal, or we can use transgenic animal for to save downstream development time for humanization or other related issues. And what we do is to harvest the spleen, the bone marrow, or the lymph nodes, depending on the immunization method, and we enrich for plasma cell, and those subset will be directly used for screening. But what we can also do is to collect blood sample from recovered patient and isolate their PBMC, and those PBMC can either be used to enrich for plasma cell that can be used directly for screening, or we can enrich for memory B cell, which will culture for several days and for B cell activation, and with those culture we can verify the ELISA positive culture, which can be pulled together for screening. So from sample collection or blood sample collection from recovered patient, isolating PBMC to verified ELISA positive culture for successful B cell activation. This process takes about 3 to 5 days, but this is significantly shorter than the 2 to 8 weeks immunization process that's required for our workflow with immunized animals. And here to present you some of our newest results for human B cell activation and single cell screening. And what you can see is that this is the B cell subset that we enriched for from frozen human PBMC. And then at day 1, we're screening, or we're staining for the CD38 marker for plasma cell, or for human plasma cell, or plasmablast. And at day 3, you can see there's a significant differentiation or expansion of the culture, or the subsets that are now CD38 positive. And at day 5, you have even a more even greater increase of that population. And for this particular test that we did at day 3, we collected the sample, and then the total cell that we used for screening was about 4 times 10 to the 5th, and the single B cell, or the total cell that we collected from the B cell activation culture is about 4 times 10 to the 5th, and the number of cells that we used for screening is about 8,500, and among those we were able to identify over 2,000 or 2,200 that are IgG positive. To summarize, the GenScript ProBio offers diverse functional leads against COVID-19 generated from different platforms, including the human naive and the llama naive for SDAB generation, or the single domain antibody generation. And the ProSpeed single B cell screening can really accelerate your antibody discovery, because first of all, it enables rapid screening, which can be done in less than a day, and it allows functional assay during the initial screening phase to save time and effort, and also money for your downstream development process. And lastly, we can provide the option to screen human B cell from recovered patients as well. So with that, I would like to thank you for being here today, and I'm going to take some questions now. So first of all, the first question is the number of cells that are screened per chip. For the screening is typically, we have different formats for the chip, and then the one that has the largest capacity is the 14K chip, which has about 14,000 nanopens, and with that we can typically pan or screen for 10,000 cells per chip. What is our sequencing success for single cell sequencing? Right now, we are looking at the single cell sequencing is never 100% successful, and right now we're looking at anywhere from 60 to 80% success rate. Is there any difference between sequencing the human B cell and the immunized animals? The answer is yes and no. Of course, this primers that are going to be different because it's different species. But in terms of the cDNA synthesis, which is the most critical step for single cell sequencing, the success rate is about the same. And then also we are doing some validation and optimization related to sequencing human B cell, but we believe that the sequencing success might even be a little bit higher than transgenic or especially transgenic animals or mouse, because the C region for human VHVL sequences are even more conserved.